This video is about a group of treatments being developed known as the Targeted Protein Degraders. At the moment, the targeted therapies we have mostly either target cell surface proteins on cancer cells, and they are some kind of antibody-based treatment, or they block a kinase enzyme, and mostly these are small molecules that get into that ATP binding site of kinases and they block kinase enzymes. We have a few other drugs that can block things that aren't on the cell surface and that aren't kinase enzymes, but it's really just a handful. Most of the things that control cancer cell behavior are things that we cannot block because they're not a cell surface protein and they're not a kinase. You might be surprised to know that I'm now going to say a bit about thalidomide. Thalidomide is a drug that was first developed as a morning sickness aid for pregnant women, but it was discovered to cause birth defects and we had to stop using it. But it never completely went away. It was developed as a treatment for leprosy, it was developed as a treatment for some skin conditions and mouth ulcers, and it was developed as a treatment for myeloma, a form of hematological cancer, and it's also now given as a treatment for patients with some forms of non-Hodgkin lymphoma. It took us a long time to work out what thalidomide does, but we now know that thalidomide targets a protein in our cells called cereblon. To understand cereblon, I need to explain proteasomes. Proteasomes are our cells' recycling machinery. Our cells are constantly making proteins and they're constantly needing to break down proteins. And to break down proteins, they mostly use their proteasomes. Every cell has roughly 1 million proteasomes. They're really important to our cells. They enable them to chop up and destroy old proteins, recycle their contents, and reuse them to make new proteins. In order for a cell to know this is a protein you want to destroy, well, that protein first has to be labeled with multiple copies of a tiny little protein called ubiquitin. And there's a group of enzymes known as the E3 ubiquitin ligases, which attach ubiquitin to proteins, telling the proteasomes this is a protein you want to destroy. Cereblon is one of these E3 ubiquitin ligases. It's just one example. There's over 700 different E3 ubiquitin ligases in our cells. Cereblon is just one of them. We can group them into 4 families, and they attach ubiquitin to thousands of different proteins, telling proteasomes to destroy them. And what cereblon does is that it tells a cell to destroy a whole raft of proteins. But if you've exposed that cell to thalidomide, it changes what proteins cereblon will attach ubiquitin to. Thalidomide creates an interface between cereblon and 2 proteins it wouldn't normally destroy, Ikaros and Ilos. Because of thalidomide, cereblon attaches ubiquitins to these 2 proteins, the cell destroys them, and that's what is responsible for all the effects, good and bad, of thalidomide. Because it works in this way, creating an interface, creating a glue between cereblon and protein targets, we call thalidomide a molecular glue degrader. There's another way to target cereblon towards new proteins, and that's to develop a chimeric protein, a mix and match protein that contains a binding site for cereblon and a binding site for the protein you want to destroy. These PROTACs, as they are called, are bulkier, but they tether those 2 proteins together, and again you can use them to destroy proteins of your choosing. So we have these 2 groups of treatments, the PROTACs and the molecular glue degraders, that we can use to destroy proteins we've identified as being important to cancer cells, and that hopefully, if we persuade the cells to destroy them, that will kill the person's cancer cells. They have different properties, the molecular glues and the PROTACs. The molecular glues, they are smaller in size. That means they distribute through the body easier. They can also be an oral treatment. The PROTACs, easier to design. We can design them piece by piece, but you end up with a bulkier compound that it's harder for it to get through the body, and it's also unlikely to be possible to create it as an oral treatment. I've given you 2 examples here of PROTACs that are in development as treatments for hormone-sensitive breast cancer and hormone-sensitive prostate cancer. The creation of these targeted protein degraders opens up a whole new world of potential targets for targeted therapies. They open up a world where we don't just have to target things that are on the cell surface or kinase enzymes. There are hundreds of E3 ubiquitin ligases that we could potentially use to create treatments that selectively destroy a whole raft of different proteins. So we're just at the start of this journey at the moment. We're just seeing the first few enter into clinical trials. The ones furthest few trials at the moment are for hormone-sensitive prostate cancer and hormone-sensitive breast cancer, but many others in development.